Hello, friend. Welcome to Science for Sleep. Whether you have returned for another quiet evening or have only just discovered this channel, this is a gentle place to slow down, learn something new, and allow the day to gradually fall away. Tonight, we are exploring chili peppers. You may know chili peppers as small red fruits. Yes, fruits. Dried flakes in a glass jar, or perhaps as part of the name of a rather famous band. But tonight, the peppers themselves are taking the stage. A fresh chili can look almost harmless. Its skin may be smooth and glossy, reflecting the kitchen light like polished wax. It may be bright red, deep green, golden yellow, or almost purple. Its surface gives no warning of the sensation hidden inside. You can hold it in your hand without feeling heat. You can place it beside a thermometer, and it may be no warmer than the tomato or cucumber next to it. Then you take a bite. At first, there may be only a crisp snap and a fresh, slightly grassy flavor. A few seconds later, warmth begins to gather along the tongue. It reaches the lips, spreads across the roof of the mouth, and may continue growing long after the pepper has been swallowed. The pepper has not raised the temperature of your mouth to the level of a hot drink. There is no flame, no glowing ember, and no true fire. Yet the nervous system begins reacting, as though something dangerously hot has arrived. How was nature able to develop a plant and a fruit that can feel so hot without actually being hot? That question will carry us deep inside the pepper, into the cells that make its chemicals, the nerves that detect them, and the long evolutionary history that shaped the burn. Chili heat comes mainly from a chemical called capsaicin. Capsaicin is almost colorless and nearly odorless. You cannot judge exactly how much is present by looking at the surface of a fruit. A small, pale pepper may contain far more of it than a large, bright red one. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. What makes capsaicin especially interesting is that it is not detected in the same way as sweetness or saltiness. The human tongue can recognize several basic tastes, including sweet, salty, sour, bitter, and umami, the savory taste found in foods such as mushrooms, tomatoes, and aged cheese. Spiciness belongs to a different sensory system. Scientists call this system chemesthesis. Chemesthesis simply means that certain chemicals can trigger nerves that normally respond to physical conditions such as heat, cold, pressure, or irritation. Menthol uses this system when it makes mint feel cool. The carbon dioxide in a fizzy drink uses it when the bubbles seem to sting. Capsaicin uses it when a chili feels hot. The nerve endings in the mouth do not taste a flame. They receive a chemical signal that resembles the warning produced by very high temperature. The reaction is real, even though the heat is not. That is why the eyes may water. It is why the nose may begin to run, and why tiny beads of sweat can appear along the forehead. The body trusts the warning sent by its nerves and begins preparing to cool and protect itself. This unusual ability is found within the genus called capsicum, the botanical group containing chili peppers. A sweet bell pepper and an extremely hot habanero, belong to this same broad group. One may contain almost no noticeable capsaicin, while the other can produce an intense burning sensation from a very small bite. That difference is not caused by the fruit's temperature. It comes from its genes, its internal anatomy, and the chemical work performed while the pepper is growing. Every glossy fruit is therefore a small biological machine. It begins as a flower. It forms walls, chambers, pale inner tissue, and seeds. Within particular cells, ordinary plant materials are joined together to build molecules capable of activating a human warning system. Those molecules may help protect the pepper's future seeds from certain animals and even from some harmful fungi. Other animals, especially many birds, can eat the fruit with little concern for its pungency and carry the seeds into new places. Humans did something even more unusual. Instead of avoiding the warning, people learned to enjoy it. They saved the seeds, grew hotter and milder plants, carried peppers across oceans, and eventually created varieties ranging from gentle paprika to fruits measured in millions of Scoville heat units. All of that history is resting quietly inside something small enough to fit in the palm of a hand. A cool fruit can awaken the nerves of heat. A plant defense can become a favorite flavor. And a chemical warning can become part of meals eaten across nearly every part of the world. That small fruit belongs to a much larger plant group than its size suggests. Walk through a market stall, and the range appears almost impossible to fit beneath one name. There may be broad green bell peppers with thick, crisp walls. Nearby, slender cayennes curve like narrow red fingers, while jalapenos sit shorter and darker, with smooth skin and blunt ends. Poblanos widen into deep green triangles. Habaneros often appear smaller, folded, and lantern-shaped. Farther south in the Andes, rocotos can resemble small apples, sometimes with dark purple or nearly black seeds hidden inside. All of these are called peppers, yet they differ in size, color, aroma, wall thickness, and pungency. Botanists place true chili peppers in the genus Capsicum. A genus is a group of closely related species that share an evolutionary history and many basic features. Capsicum belongs to the nightshade family, known to scientists as Solanaceae. The same family also contains tomatoes, potatoes, eggplants, and tobacco. Once a pepper flower is pollinated, the swollen base of the flower begins developing into the part we eat. That makes a pepper a fruit in botanical terms because it grows from a flower and encloses seeds. Humans have domesticated 5 major capsicum species, although their relationships are more tangled than 5 completely separate branches. The most familiar is Capsicum annuum. Its name can sound as though it describes an annual plant, yet many members can live for several years in warm climates without frost. This one species includes sweet bell peppers, jalapenos, poblanos, many paprika peppers, and numerous cayenne-type varieties. A thick-walled bell pepper and a narrow, pungent cayenne may look unrelated, though generations of selection shaped both from the same species. Capsicum chinense contains habaneros and many peppers bred for extreme heat. The name chinense came from an 18th century mistake. European botanists encountered these peppers in collections associated with Asia and assumed they came from China, even though every capsicum species originated in the Americas. Many Chinense peppers carry strong fruity or floral aromas beneath their pungency. Their fruits can form lanterns, flattened bonnets, rounded pods, or deeply folded shapes. Capsicum frutescens often produces smaller, upright fruits. Tabasco peppers are a familiar example, with pods that point above the leaves and change from pale green through yellow and orange before becoming red. Capsicum baccatum is closely tied to South American ají peppers. Several of its flowers show yellow or green markings near the center, and its fruits often carry bright, citrus-like aromas. Farmers in Peru, Bolivia, and nearby regions have maintained many local forms. The fifth domesticated species, Capsicum pubescens, developed along a different path in the Andean highlands. Its leaves and stems may carry fine hairs, which explains the name pubescens, meaning hairy. Rocoto fruits from this species tolerate cooler conditions better than most tropical chilies. Their seeds are commonly dark, providing an easy clue when the fruit is cut open. These names help scientists describe broad patterns, although cultivated peppers have crossed, moved, and exchanged genes over long periods. Gene flow happens when pollen or seeds carry inherited material from one population into another. Farmers also moved plants between regions, selected unusual offspring, and sometimes allowed related types to cross. Modern genome studies can detect traces of those old exchanges within the plant's DNA. A family tree of peppers can therefore include branches that touch and share inherited material. Selection widened the visible differences even further. People saved seeds from fruits that were larger, sweeter, easier to dry, better suited to cool mountains, or more pungent. Repeated over hundreds of growing seasons, those choices could transform fruit size, shape, color, and chemistry while leaving the plants closely related underneath. The market basket is a display of what variation and selection can produce within one plant lineage. Across all 5 domesticated species, the fruits still grow around a shared internal plan. Beneath the colored outer wall are hollow spaces, pale supporting tissue, and seeds attached near the center. That pale tissue is where nearly all of the chemical heat begins. A clean slice through a ripe pepper reveals a structure that is much easier to understand than its heat first suggests. The knife passes through the glossy skin with a soft crack, then through the crisp flesh beneath it. Inside, the fruit opens into hollow chambers divided by pale ribs. Clusters of flat seeds cling near the center, while the colored outer wall forms most of the part people usually eat. Botanists call that outer wall the pericarp. The word simply means the wall of the fruit. In a bell pepper, the pericarp can become thick, juicy, and firm enough to hold its shape when sliced. In many slender drying chilies, it remains much thinner, allowing water to leave the fruit more quickly after harvest. The pale tissue running through the center has a different job. This tissue is called the placenta. It supports the developing seeds and connects them to the fruit while they grow. The name may sound unusual in a plant, but the basic idea is simple. It is the structure that holds and feeds the seeds during development. This is also the main place where capsaicinoids are produced. Capsaicinoids are the family of chemicals responsible for chili pungency. Capsaicin is the best-known member of that family. though several related compounds can add to the final sensation. The strongest concentrations usually gather in glandular cells associated with the placental tissue. These cells are tiny chemical workshops built into the pale ribs and central core. The seeds themselves usually do not make the capsaicin. They become pungent because they sit against the placenta and can be coated by material released from nearby tissue. That contact created the long-running belief that the seeds are the hottest part of the pepper. A person can remove every loose seed from a sliced chili and still leave behind most of the burn. The pale ribs often hold far more capsaicinoid material than the seeds they support. Removing those ribs usually reduces pungency, much more than shaking out the seeds alone. The difference can be felt in a single fruit. A small piece cut only from the colored outer wall may taste fresh, grassy, or slightly sweet, before a mild warmth appears. A piece that includes the pale inner tissue can produce a much stronger sensation, even when both pieces came from the same pepper. The pattern becomes less tidy in very pungent cultivars. In peppers such as habaneros, Carolina reapers, and other highly pungent forms, capsaicinoids can also spread into nearby inner wall tissue. The placenta may expand across folded surfaces and reach farther around the fruit's hollow chambers. Some superhot peppers have deeply wrinkled interiors with a large area of pale tissue, compared with their overall size. That gives more space for capsaicinoid-producing cells to develop. The colored wall still performs several other jobs. It protects the seeds from drying too quickly. It stores water, sugars, acids, pigments, and aroma compounds. As the fruit ripens, green chlorophyll may fade, while red, orange, or yellow pigments become more visible. Those colors help make ripe fruit noticeable to animals that can carry the seeds away. The placenta remains connected to that larger system. It supports the seeds, receives sugars and other materials from the plant, and houses much of the chemistry behind pungency. The hollow space around it also gives the seeds room to mature, without being packed into solid flesh. A pepper therefore divides its work among several tissues. The outer wall handles protection, storage, color, and much of the edible texture. The placenta supports the seeds and contains the main capsaicinoid-producing cells. The seeds carry the next generation, protected inside the fruit until they are mature enough to begin another plant. Seen from the outside, a pepper looks like one simple object. Inside, its tissues carry out separate tasks that depend on one another. The burn can now be placed in a real location. It begins mainly along the pale inner structure, within living cells too small to see without magnification. Inside those cells, plant sugars and other raw materials enter a chain of chemical reactions. Enzymes guide each step, while genes provide the instructions that allow the reactions to happen. Before capsaicin can reach a human nerve, the pepper must first build it molecule by molecule. Early in a pepper's growth, the fruit is still small, green, and chemically unfinished. Its seeds are developing, its walls are expanding, and the pale placental tissue is beginning to carry out more specialized work. Capsaicin does not arrive inside the seed as a complete substance. Placental cells manufacture it gradually as the fruit develops. The process depends on a chain of chemical reactions known as a biosynthetic pathway. A biosynthetic pathway is simply a sequence in which cells turn basic materials into a more complex molecule. Each step is guided by an enzyme. Enzymes are proteins that help particular chemical reactions happen more quickly and in the correct order. The pepper builds capsaicinoids through two main chemical routes that begin separately. One route belongs to a larger system called the phenylpropanoid pathway. Plants use this pathway to make many compounds involved in structure, color, scent, and defense. In chili peppers, it supplies the vanilla portion of the capsaicin molecule. This is the part related to vanillin, the aromatic compound associated with vanilla, although capsaicin itself does not smell like vanilla. The second route produces a branched fatty acid chain. A fatty acid chain is a line of carbon atoms joined together, somewhat like a short, flexible tail. The exact shape of this tail helps determine which capsaicinoid is formed and how strongly it interacts with sensory receptors. These two branches operate through several stages inside the placental cells. Raw materials are altered, moved, and prepared until the two finished portions can be joined. A final group of enzymes brings them together. The result is capsaicin, or one of several closely related capsaicinoids. Capsaicin is therefore part of a chemical family. Dihydrocapsaicin is another major member. In many pungent cultivars, capsaicin and dihydrocapsaicin together account for more than 90% of the total capsaicinoids. Smaller amounts of compounds such as nordihydrocapsaicin and homocapsaicin add slightly different forms of pungency. The balance among them helps shape how quickly a burn appears and how long it seems to remain. Genes provide the instructions for the enzymes that carry out this work. One especially important gene is called PUN1. PUN1 gives the plant instructions for making an acyltransferase, an enzyme involved near the final stage of capsaicin assembly. When PUN1 functions normally, the two chemical branches can be joined efficiently. Certain inherited changes can interrupt that step and greatly reduce pungency. This helps explain how a sweet bell pepper can share much of its ancestry with a hot cayenne while producing almost no noticeable capsaicin. Another important gene is called PAMT. It helps prepare material in the vanillyl branch of the pathway. Lower activity or disruptive forms of PAMT can redirect the chemistry toward other compounds, leaving less material available for capsaicin production. The pathway includes additional genes as well. PUN3 influences the movement or accumulation of materials needed for pungency. A gene called CAKR1 affects another step, connected to the fatty acid side of the process. Researchers continue to study how these genes work together, because pungency depends on a network rather than a single switch. Fruit development also changes the output. Capsaicinoid production often rises as the pepper grows, reaches a high point during a particular stage of ripening, and may later level off or decline. The timing differs among cultivars. Temperature, water supply, plant health, and available nutrients can influence the amount that finally accumulates. Two peppers carrying similar genes can therefore finish with different concentrations. Inside each placental cell, the final result comes from inherited instructions, working under the conditions of one growing season. A bell pepper carries a pathway with crucial steps reduced or interrupted. A jalapeno runs the pathway at a moderate level. A habanero can fill its placental tissue with far greater amounts of capsaicinoids. The visible fruit gives few clues about the detailed chemistry happening inside. Once capsaicin has been assembled, it remains an ordinary molecule resting in plant tissue. It has no flame, no stored temperature, and no sensation of its own. Its heat begins only after the molecule reaches a living nerve and fits into a microscopic gate built to respond. After a bite, capsaicin leaves the pepper tissue and spreads through the thin layer of moisture covering the tongue and lips. Some molecules settle against nerve endings positioned just beneath the surface. These sensory nerves are especially dense in areas that need fast protection, including the mouth, nose, eyes, and skin. Their job is to detect conditions that might damage tissue and send an early warning toward the brain. Capsaicin reaches a microscopic structure built into the outer wall of certain nerve cells. Its full name is transient receptor potential vanilloid 1, usually shortened to TRPV1. A receptor is a structure that responds when the right chemical or physical condition reaches it. This particular receptor acts as a tiny gate. When the gate is closed, charged particles remain separated across the nerve cell's outer membrane. When capsaicin attaches to the receptor, the protein changes shape and the gate opens. Sodium and calcium ions can then flow into the cell. An ion is an atom or molecule carrying an electrical charge. The movement of millions of these charged particles changes the electrical balance across the nerve membrane. Once that change becomes strong enough, the neuron produces a rapid electrical impulse. That impulse travels along a narrow nerve fiber toward the spinal cord and then into the brain. The full sequence unfolds quickly. A capsaicin molecule reaches the receptor. The gate opens. Ions enter. And an electrical warning begins moving through the nervous system. The same heat-sensitive channel responds to more than chili peppers. Researchers describe TRPV1 as polymodal, meaning it can be activated by several different kinds of stimulus. Acidic conditions can open it. Inflammatory chemicals released near injured tissue can make it more sensitive. Potentially damaging physical heat can activate it as well. In controlled laboratory conditions, many TRPV1 channels begin responding strongly near 43 degrees Celsius. That temperature is hot enough to threaten living tissue if exposure continues. The exact threshold is not fixed in every situation. Inflammation can lower it, allowing warmth that would normally feel comfortable to become painful around an injury. Cell chemistry and previous stimulation can also change how readily the gate opens. Capsaicin fits into this existing warning system. The electrical impulse produced by the nerve does not arrive in the brain with a separate label identifying the molecule. It travels through pathways already used for burning heat, acid, and chemical irritation. The brain receives the pattern of activity and produces the familiar experience of a hot, stinging mouth. This explains why the sensation feels so convincing. The heat-sensitive channel is genuinely active. The nerve is carrying a real electrical signal. The brain is responding to a pathway normally connected with physical danger. Only the original trigger is different. A spoonful of hot soup activates these nerves because thermal energy warms the tissue. A chili activates many of the same nerves because capsaicin binds directly to their receptors. Both routes can lead toward a similar perception. The mouth can even receive both signals at once. A steaming curry containing chili may activate TRPV1 through high temperature and capsaicin together, making the first bite feel especially intense. Cool food can carry a strong capsaicin signal without adding thermal heat. This receptor is found in small sensory neurons known as nociceptors. The word nociceptor refers to a nerve cell that detects potentially harmful conditions. Their endings branch through exposed tissues, much like fine roots spreading through soil. One branch may lie close to the surface of the tongue, while another reaches the lining of the throat. Related nerves are present around the eyes, where even a tiny transferred amount of capsaicin can produce a powerful warning. The signal grows as more receptors open and more neurons begin firing. A mild pepper activates a smaller response. A concentrated patch of placental tissue can expose many nearby endings at once. The resulting activity spreads through networks that connect sensation with automatic protective responses. Saliva begins to gather. Blood vessels near the skin may widen. Sweat glands can receive instructions even though the body's internal temperature remains within its usual range. By the time the burning sensation has fully formed, the message has already traveled well beyond the surface of the tongue. From the mouth, the warning begins moving through systems that control far more than conscious sensation. A strong chili may leave the tongue burning, while the forehead grows damp. The nose can begin to run, and the eyes may fill with tears even though the pepper never touched them. These reactions are connected by nerves that help protect exposed parts of the body. Much of the irritation from the face, mouth, and nose travels through the trigeminal nerve. This large nerve divides into several branches across the face. Its sensory endings help detect pressure, temperature, and irritating chemicals. When capsaicin activates those endings, the signal reaches brain regions that organize protective responses. Saliva is one of the first visible results. Glands beneath the tongue and around the jaw release more fluid into the mouth. That extra moisture can dilute irritating material and help move it away from sensitive tissue. Swallowing also becomes more frequent, carrying some of the capsaicin toward the stomach. The nose responds in a similar way. Glands in the nasal lining release watery fluid when nearby sensory nerves report chemical irritation. This is why a pungent meal can produce a runny nose even when no infection or allergy is present. The fluid helps wash the lining and carry particles toward the throat or nostrils. Tears are part of the same general defense. Signals from facial sensory nerves can activate the lacrimal glands located above the outer corners of the eyes. Tear fluid spreads across the surface each time the eyelids close. That response is especially strong if capsaicin reaches the eye directly from a fingertip, cooking vapor, or a tiny droplet. Sweating comes from a different branch of the nervous system. The autonomic nervous system controls actions that usually happen without conscious effort, including sweating and changes in blood vessel width. When the brain receives a strong heat-like warning from the mouth, sweat glands around the forehead, scalp, and upper lip may become active. This is called gustatory sweating because it is triggered by eating. The sweat evaporates from the skin and carries away heat, following the same basic cooling process used during exercise or warm weather. blood vessels close to the surface may widen at the same time. Wider vessels allow more warm blood to pass near the skin, sometimes producing visible redness across the cheeks or ears. The body is using a familiar heat control program in response to sensory information from capsaicin. Core temperature usually remains within its normal range during an ordinary spicy meal. The nervous system is responding to the pattern arriving from heat-sensitive channels, rather than waiting for the entire body to become dangerously warm. Capsaicin can also continue activating nerves after it leaves the mouth. Heat-sensitive channels occur in the throat and along parts of the airway. Fine chili particles in cooking smoke can produce coughing, because sensory nerves detect them as irritants. After swallowing, capsaicin travels through the esophagus and into the stomach. Related receptors are found throughout the digestive tract, including sections of the small and large intestine. This wider receptor map helps explain why the sensation may continue long after the flavor of the pepper has faded. The strength of the response differs greatly from one person to another. Dose matters because a concentrated piece of placental tissue exposes more receptors than a thin slice of mild pepper. Previous experience can reduce the intensity a person reports, while sensitive tissues may react strongly to a small amount. Expectation also shapes attention. Someone familiar with chili heat may recognize the rising pattern and remain calm, while an unexpected bite can feel sharper because attention immediately locks onto it. Even so, the physical responses are genuine. Nerves release signaling chemicals. Glands produce fluid. Blood flow near the skin changes, and sweat glands begin working. These events consume very little energy compared with exercise and do not mean that a spicy meal is rapidly burning away large numbers of calories. Their purpose is protection and temperature control. Each tear, flush, and bead of sweat follows a coordinated pathway built to respond to possible harm. The body then tries to clear the molecule that started the signal. A drink of plain water often brings only brief relief, because capsaicin does not mix with water as easily as it mixes with fats and cell membranes. The first instinct is often to reach for a cold glass of water. For a few seconds, the cool liquid lowers the temperature at the surface of the tongue and lips. That can soften the sensation briefly. Once the water is swallowed, the burning feeling often returns. Capsaicin remains behind because it has very low solubility in water. Solubility describes how easily one substance spreads evenly through another. Sugar dissolves readily in water because its molecules interact well with water molecules. Capsaicin behaves differently. Part of its structure is drawn toward lipids, the fats and oils found in food and in the membrane surrounding living cells. Chemists describe this property as lipophilic, which means fat-loving. A capsaicin molecule can settle into oily food, cling to biological membranes, and remain close to heat-sensitive nerve endings. Plain water has difficulty pulling it away. The movement of water can sometimes spread the irritation across a larger surface. A sip may carry tiny droplets or food particles from the center of the tongue toward the lips, gums, or roof of the mouth. The total amount of capsaicin has not increased, though more tissue may now come into contact with it. This helps explain why the sensation can seem to widen after drinking. Cold water still provides real temperature relief, Cooling the tissue can reduce the activity of heat-sensitive nerves for a short time. Chemical removal is a separate process. When the cold liquid leaves, capsaicin may still be resting in the same membranes and reopening the same channels. Foods containing fat behave differently because capsaicin can move into another oily substance more easily than it moves into water. Milk contains water along with fat droplets, proteins, and dissolved sugars. Those components create several ways to dilute or carry away material from the mouth. Casein, one of the major proteins in milk, can interact with oily substances and help disperse them through liquid. The physical volume of the drink also washes loose particles away. Milk is therefore more useful than plain water in many situations, although its effect varies with fat content, temperature, the amount of capsaicin, and the tissue already exposed. Other solid foods can reduce the concentration by mixing capsaicin into a larger mass. Bread, rice, or another mild food may pick up oily material from the mouth as it is chewed. Food containing fat can provide an additional place for the molecule to settle. These effects are gradual and incomplete because capsaicin may already be embedded near nerve endings. Its ability to cling also explains why handling chilies can produce a delayed surprise. A small amount may remain on the fingertips after cutting a jalapeno or habanero. Touching the eyelid later transfers the molecule to tissue packed with sensitive nerve endings. The amount can be too small to see and still produce strong irritation. Soap works better than water alone because soap molecules have one end that interacts with water and another that interacts with oils. They can surround oily material and help carry it away during rinsing. Inside the mouth, the sensation also fades through changes in the nerves themselves. Continued activation allows calcium ions to enter the sensory neuron. That rising calcium level helps trigger processes that make the nerve less responsive for a time. The heat-sensitive channels may open less readily, and the nerve ending may release fewer signaling chemicals. This short-term reduction is called desensitization. It can begin during a single meal. The burn rises as more receptors become active, reaches a peak, and then weakens as capsaicin is diluted, swallowed, moved away, and met by nerves that are responding less strongly. 3 separate processes are therefore unfolding together. Some capsaicin is physically cleared from the tissue. Cooling changes the sensation for as long as the temperature remains lower. Neural desensitization reduces the response even when small amounts of the molecule are still present. Their timing differs, which is why relief can arrive in waves rather than all at once. The nervous system gradually returns toward its earlier state after the exposure ends. Repeated contact over many meals can extend that change beyond a few minutes, allowing the same pepper to feel quite different to someone who has learned its burn over time. At the same table, 2 people can take the same spoonful of chili stew and have very different experiences. One may notice a sharp burn that takes over the whole meal. The other may first detect roasted tomato, smoke, sweetness, and only a manageable layer of heat beneath them. The capsaicin concentration in the bowl is the same for both people. Their sensory systems and histories are not. Some people are naturally more sensitive to capsaicin than others. Differences in nerve response, mouth tissue, attention, and previous exposure all influence how strong the burn feels. A person who rarely eats chili may notice every change as the heat spreads across the tongue. Someone who has eaten pungent food for years may still activate the same heat-sensitive channels, yet the experience can feel less threatening and easier to place within the rest of the flavor. Repeated exposure does not remove TRPV1 receptors from the mouth. Those receptors remain part of the sensory system. The change comes from several processes working together. During one meal, active nerves can become less responsive for a time. Across many meals, the person also becomes familiar with the pattern of the sensation. The burn rises after the first bite. It reaches a level that can be recognized. Then it gradually fades. That pattern becomes predictable. Prediction matters because the brain pays close attention to unexpected signals. An unfamiliar burning sensation can pull nearly all attention toward itself. A familiar one leaves more space for the rest of the food. Expectation does not stop capsaicin from opening the receptor. It changes how the signal is understood. The person knows the source, recognizes the likely duration, and has learned that an ordinary serving will pass without injury. Researchers sometimes describe chili enjoyment as a controlled aversive experience. Aversive means naturally unpleasant or worth avoiding. People can learn to enjoy a controlled version of such a sensation when its limits are clear. A roller coaster can create fear within a known structure. A very sour candy can produce a strong facial reaction while remaining safe to eat. Chili heat can work in a similar way. The warning is intense enough to feel vivid, while the meal provides a familiar setting and a known endpoint. Cultural learning often begins with small amounts. A child may first taste a mild sauce mixed into rice or stew. Later meals may contain slightly more. Family recipes establish the expected level of heat, and repeated exposure teaches which sensations belong with which foods. In regions where pungent dishes are common, tolerance may develop gradually over years rather than through any deliberate attempt to train it. Flavor also changes the meaning of the burn. A smoked pepper carries compounds formed as wood burns, and its smoke settles on the fruit. A ripe habanero may contribute floral and tropical aromas. Fermented chili paste can contain acids and new aroma compounds produced by microbes during storage. Salt and sweetness can soften or reshape the way pungency is noticed. These qualities arrive together in the mouth, so the experience becomes connected to a whole dish rather than to capsaicin alone. Memory adds another layer. The smell of a familiar curry, hot sauce, or pepper stew can bring back meals shared many times before. Expectation begins before the first bite, guided by color, aroma, and the sight of the food. By the time capsaicin reaches the nerves, the listener's body already has a sense of what is coming. Endorphins are sometimes offered as the single explanation for why people enjoy chili. The full picture is broader. The nervous system can release several signaling chemicals during strong sensory stimulation, though enjoyment also depends on learning, culture, flavor, and personal preference. Capsaicin is not chemically addictive in the way nicotine or certain drugs are. People may crave a favorite spicy meal because they enjoy its combined sensations and associations. Humans eventually carried this preference far beyond the dinner table. They saved seeds from peppers with useful levels of heat. Farmers planted those seeds again, traded them between communities, and selected fruits with particular aromas or shapes. Over generations, people maintained some peppers for gentle warmth and pushed others toward much greater pungency. A chemical that once discouraged sensitive mammals became a trait humans actively preserved. That reversal only makes sense when the pepper's original audience comes back into view. The burn had value because certain animals damaged the seeds, while others could carry them safely into new ground. Long before people planted chilies in gardens, small wild fruits ripened among shrubs in the Americas. Their bright color made them easy to notice against green leaves. Soft pulp offered moisture and energy, while each fruit carried seeds that needed to leave the parent plant. For many fleshy fruits, dispersal is the main reason the edible tissue exists. An animal eats the fruit, moves across the landscape, and later releases the seeds somewhere new. Distance can improve a seed's chances. Directly beneath the parent, young plants compete with established roots for water and minerals. The shaded ground may offer too little light. Fungi and insects that already gather around the adult plant can also find nearby seeds more easily. A seed carried several hundred meters away may reach open soil with less competition and a different collection of enemies. The fruit therefore benefits from being eaten by the right visitor. Its sweetness attracts animals, although every eater does not provide the same service. Small mammals often hold food between their front paws and break it apart with sharp teeth. Rodents may chew the fruit wall, crush the seeds, or collect those seeds as food. A chili seed damaged between mammalian teeth has little chance of becoming a new plant. This creates a difficult balance. The ripe fruit needs to be noticeable and rewarding enough to attract a carrier. Its seeds also need protection from animals that consume them without moving them safely. Capsaicin helps create that filter. Mammals usually possess heat-sensitive channels that respond strongly when capsaicin reaches the mouth. A wild chili can therefore feel painfully pungent to a mouse or other small mammal, even though its pulp contains useful water and sugar. Experiments with wild peppers have shown that some mammals reduce their feeding when capsaicin is present. They may sample the fruit and leave, or choose a less pungent fruit nearby. That avoidance can protect a larger share of the seeds from crushing. Biologists sometimes describe this pattern as directed deterrence. The phrase means that a defense discourages consumers that cause the greatest harm, while leaving access open to animals that provide a useful service. The plant does not need to make its fruit unpleasant to every creature. A universal deterrent would leave ripe fruit hanging until it's spoiled or dropped beside the parent. Capsaicin works more narrowly because animal sensory systems differ. Over many generations, plants that produced an effective amount of the chemical could leave more surviving offspring under certain conditions. Their seeds escaped damaging mouths more often, while useful dispersers continued feeding. Those surviving plants passed the genes for pungency into later generations. This process is called differential reproduction. Individuals with a helpful inherited trait produce more successful descendants than individuals lacking it. The result can gradually reshape a population without planning or intention. Local conditions still matter. Where seed-eating mammals are common, strong deterrents may provide a clear advantage. In places with fewer harmful consumers, producing large amounts of capsaicin may offer less benefit. Chemical defenses require energy and raw materials that could otherwise support growth, flowers, or additional fruit. Wild populations can therefore contain peppers with different levels of pungency. The trait reflects a balance among cost, protection, and the animals present in that habitat. A small red fruit on a wild shrub is carrying out several jobs at once. Its color advertises ripeness. The pulp rewards an animal willing to swallow it. Capsaicin changes which visitors are likely to continue feeding. Human mouths experience that system as heat, though its older consequence was measured in intact seeds and successful seedlings. The strategy depends on an animal capable of seeing the red fruit, swallowing it with little damage, and carrying the seeds away. Many birds can do exactly that because the capsaicin warning reaches their sensory nerves very differently. A small bird landing on a wild chili shrub may pluck a ripe red fruit and swallow it in one quick motion. The fruit disappears whole, with very little chewing. That simple feeding style protects the seeds far better than the jaws of a rodent. A mouse may hold the same fruit between its front paws, bite through the wall, and grind the contents with its teeth. The bird often carries the seeds through its digestive system, with much less physical damage. Its ability to ignore the pungency begins inside the sensory nerves. Birds have their own version of the TRPV1 heat-sensitive channel. That channel still performs an important protective job. It responds when tissue becomes dangerously warm, and it can react to acidic conditions. Birds therefore feel real heat and can move away from surfaces or foods that might injure them. Capsaicin interacts with the avian channel much less strongly. Studies using chicken TRPV1 have shown that small differences in the receptor's structure alter the way capsaicin fits into it. A few changes in the arrangement of amino acids, the small building blocks of proteins, can make the channel far less responsive to the chili molecule. The gate still opens for harmful temperature. Capsaicin has much more difficulty opening it. A wild chili can therefore produce two very different experiences from the same fruit. A mammal receives a strong warning through capsaicin-sensitive nerve endings. Many birds encounter the fruit with little or no noticeable pungency. Sensitivity varies among bird species, so the effect is not perfectly identical across every feathered visitor. The broad pattern is still strong enough to influence which animals feed. Wild chilies often produce fruits much smaller than the peppers found in grocery stores. Some are only about the size of a pea. Their red color stands out clearly against green leaves, especially to birds searching for ripe fruit. The small size also makes swallowing easy. Once inside the bird, the soft pulp is broken down, and the seeds travel through the digestive tract. Their hard outer coats protect the living embryo within. Later, the seeds leave the bird in a new location. That journey can carry them beyond the reach of the parent plant's roots and into a different patch of soil. A bird crossing an open field or moving between forest edges can transport seeds much farther than gravity alone. Even a short flight of a few hundred meters may place a seed in a new light level, moisture pattern, and community of competing plants. The parent shrub remains behind, while its offspring gains access to another part of the landscape. Passage through the digestive system may also change what happens after the seed lands. Pulp is removed, which can reduce the scent that attracts some seed predators. The seed coat may be lightly worn by digestion, sometimes affecting how readily water enters during germination. Deposited droppings can add a small amount of organic material around the seed. These effects differ among bird species and plant populations, so gut passage does not guarantee successful growth. It changes the conditions surrounding the seed and can improve its chances in certain habitats. The entire relationship depends on a tiny molecular difference. A small variation in one receptor protein changes how an animal experiences capsaicin. That altered sensation influences which fruit it eats. Feeding behavior then affects where the seeds travel. Across many generations, those movements help shape the spread and survival of wild pepper populations. The fruit's red skin, small size, and chemical defense work together as parts of the same dispersal system. Birds gain food from the pulp. The pepper gains movement for its seeds. Mammals that might crush those seeds are more likely to turn away. Even this effective arrangement leaves the fruit exposed while it ripens on the branch. Before a bird arrives, a small insect can pierce the skin and leave an opening only a fraction of a millimeter wide. That narrow wound may be enough for fungal spores to reach the seeds inside. Through that tiny puncture, the fruit's protective skin is no longer complete. A sap-feeding insect may leave a wound so small that it is difficult to see without magnification. Many of these insects belong to the order Hemiptera, a large group that includes leafhoppers, shield bugs, and other species with narrow mouthparts built for piercing plant tissue. The mouthpart enters like a fine, hollow needle. Sap, or fruit juices, provide the insect's meal, while the opening it leaves behind can become an entrance for microbes. Fungal spores are common in soil, dust, rain splash, and the surfaces of nearby plants. A spore landing on an intact chili may remain outside. Placed beside a fresh puncture, it can grow through the broken skin and reach the moist tissue within. Species in the genus Fusarium are among the fungi known to infect wild chilies. Once established, thin fungal threads spread through the fruit. These threads, called hyphae, absorb nutrients as they grow. Together, they form a larger network known as a mycelium. The infected tissue may soften, darken, or collapse, while fungal growth advances toward the seeds. A seed can appear whole from the outside and still lose its ability to germinate if the living tissue inside has been damaged. This danger was studied closely in eastern Bolivia, using a wild species called Capsicum chaquense. Its fruits are small and round, often no larger than a pea. Some plants produce pungent fruit, while nearby plants can produce fruit with little or no capsaicin. Researchers examined 7 wild populations spread across roughly 1,600 square kilometers. That broad area included places with different moisture levels, insect activity, and fungal pressure. The natural variation allowed pungent and non-pungent plants to be compared under real field conditions. Fruits carrying insect punctures were more likely to contain fungal infection. Pungent fruits generally supported much less fungal growth than non-pungent fruits growing in the same region. In the study, fungal loads in pungent fruit were about 45 to 55% lower. The pattern was strongest in locations where piercing insects and fungal damage were common. Populations facing greater pressure also contained a higher proportion of pungent plants. Field observations showed the pattern, while laboratory experiments tested the chemistry directly. Researchers placed fusarium on growth material containing concentrations of capsaicinoids similar to those measured in wild fruit. Capsaicin reduced fungal growth by about 33%. Dihydrocapsaicin, another major chili compound, reduced growth by about 16%. The two substances did not sterilize the material. They slowed the fungus enough to improve the seed's chance of surviving until dispersal. That difference can matter greatly across thousands of fruits and many generations. A plant whose seeds remain healthy slightly more often can leave more descendants than a nearby plant, losing a larger share to rot. Pungency still carries a cost. Building capsaicinoids requires carbon, energy, enzymes, and water moving through the fruit. In dry environments, plants already face limits on growth and reproduction. Studies of wild chilies suggest that pungent plants can use water less efficiently under drought conditions than non-pungent plants. Where fungal pressure is low and water is scarce, reduced pungency may therefore remain useful. The population does not settle on one perfect answer. Local conditions maintain a mixture of traits because the value of chemical defense changes from place to place. The fungi are changing as well. Later studies found chili-associated fungi with alternative respiratory pathways that help their cells continue producing energy during capsaicin exposure. Some strains can tolerate concentrations that suppress other fungi. A few can even break capsaicin down and use parts of the molecule. The defense shapes the fungus, while fungal resistance changes the value of the defense. Wild chilies developed within that moving balance of insects, microbes, animals, and climate. Their fruits remain small, thin-walled, and closely tied to the conditions of one habitat. Long before people selected large bells or long cayennes, these delicate berries were already carrying the results of countless generations of pressure across the American landscape. In the wild, a chili pepper often looks less like a grocery store vegetable and more like a bright berry held on a thin green branch. A chiltepin fruit may be only about the size of a pea. Its wall is thin, its shape is nearly round, and its color changes from green to a clear, shining red as it ripens. The fruit often points outward or upward, where a passing bird can see it easily among the leaves. This small size suits the animal carrying it. A bird can pluck the whole fruit, swallow it quickly, and move the seeds without grinding them between teeth. When the fruit reaches full ripeness, it may detach with little force. A light tug from a beak can separate it from the stem, while many cultivated peppers have been selected to remain attached until a person harvests them. Wild plants also produce fewer layers of thick, juicy flesh. Their fruits invest less in the large walls found in bell peppers, and more in a compact package that can be eaten and transported efficiently. Chiltepins still grow in parts of Mexico and the southwestern United States. They often appear along rocky canyons, dry foothills, and the shaded ground beneath larger shrubs or trees. These neighboring plants can act as nurse plants, giving young chilies some protection from intense sun and helping the soil remain moist for longer after rain. Farther south, other wild capsicum species occupy very different landscapes. Southeastern Bolivia and nearby regions contain an especially rich collection of wild peppers. Capsicum chaquense grows in seasonally dry country. Related species occur in forest edges, mountain valleys, and highland habitats where temperature and rainfall differ sharply across short distances. Mexico, Central America, the Caribbean, and much of South America contain additional wild lineages shaped by their own surroundings. No single living wild species should be treated as the one ancestor of every cultivated chili. The genus had already divided into many branches before farming began. Some populations became separated by mountains. Rivers and dry plains limited the movement of pollen and seeds. Birds still carried fruit between nearby habitats, allowing some gene flow, which means inherited material moved from one population into another. New mutations also appeared over time. A mutation is a change in DNA that can create a new version of a trait. Most changes have little effect, though some alter fruit color, flowering time, seed size, pungency, or tolerance for cold and drought. Local conditions then influence which traits remain common. A population visited by many seed-eating mammals may benefit from stronger pungency. Another living under intense fungal pressure may gain protection from higher capsaicinoid levels. In a dry valley, a plant that uses water more carefully may leave more offspring, even if its fruit is less pungent. Bird species also differ from one habitat to another, changing the size, color, and position of fruits most likely to be carried away. This is why wild diversity matters. Domestication did not begin with one uniform pepper that people reshaped from a blank starting point. Human communities encountered plants that already differed in useful ways. One shrub might carry slightly larger fruit. Another could produce a stronger aroma or ripen earlier in the season. A nearby plant might hold its fruit longer, making gathering easier. The first changes toward cultivation were probably gradual. People collected ripe chilies from familiar patches and returned to the same places in later seasons. Useful plants growing near camps or settlements may have been left undisturbed. Seeds dropped with food scraps could sprout close to where people lived. Travelers carried dried fruit or seed between valleys, placing pepper genes in landscapes they might never have reached through birds alone. Repeated gatherings slowly became deliberate planting. Once people began choosing seed from particular fruits, a new form of selection entered the pepper's history. A bird favored fruit it could see and swallow. A fungus favored tissue it could invade. Human hands favored peppers that were easier to gather, more pleasant to cook, or better suited to local soil and weather. Those choices began amplifying differences that had already been present in the wild. Once people began planting saved seeds, small differences could accumulate from one harvest to the next. A family might keep seed from the largest fruit, the earliest ripening plant, or the pepper that held firmly to its stem during gathering. Another community might prefer a stronger aroma, thicker flesh, or a level of heat suited to a familiar dish. Each choice slightly changed which plants produced the following generation. Repeated across centuries, those choices reshaped entire populations. The most widely cultivated species today is Capsicum annuum. Its domestication history is closely tied to Mexico, where several kinds of evidence point toward an important center of early cultivation. Modern genetic studies compare patterns in the DNA of wild and domesticated plants. Ecologists map where likely wild relatives still grow. Archaeologists study ancient plant remains, while language researchers trace old names for peppers through indigenous languages. Together, these lines of evidence suggest that Capsicum annuum was domesticated somewhere across northeastern to central eastern Mexico. The exact location remains difficult to reduce to a single valley or settlement, because cultivation developed gradually and plants continued moving between communities. From that broad history came an extraordinary range of peppers: bell peppers, jalapenos, poblanos, cayenne types, and many paprikas, all belong to Capsicum annuum. Their shared species hides enormous differences created through selection. Some farmers favored broad fruits with thick walls. Others retained long, narrow peppers that dried quickly in the sun. Color also mattered because green, red, yellow, orange, and darker fruits could serve different cooking or preservation traditions. Farther south, other capsicum species followed their own domesticated paths. Capsicum baccatum became an important cultivated group in South America. Its many ají peppers remain closely associated with Peru, Bolivia, and neighboring regions. Some varieties carry bright citrus-like aromas, while others were selected for particular colors, shapes, or drying qualities. Capsicum pubescens developed strongly in the cooler Andean highlands. Rocoto peppers from this species can grow where many tropical chilies struggle. Their leaves often carry fine hairs, and their dark seeds provide a clear visual difference from most cultivated peppers. Highland farmers selected plants able to flower and fruit under cooler nights and mountain conditions. Capsicum chinense became especially important in tropical parts of the Americas. Habaneros and many modern superhot peppers belong to this species. Its fruits often carry strong floral or fruity aromas alongside their pungency. Capsicum frutescens include smaller, often upright peppers, such as the type used for Tabasco sauce. The relationship between chinense and frutescens is especially complex. They are closely related, and pollen movement between cultivated or wild populations allowed genes to cross old boundaries. Modern DNA studies sometimes reveal patterns that resemble overlapping networks more than 5 perfectly isolated branches. Domestication changed several traits at the same time. Wild fruits often detach easily when ripe because birds need to remove them. Farmers generally benefit from fruit that remains attached until harvest. Cultivated peppers also became larger, with more flesh surrounding each seed. Ripening could become more synchronized, allowing many fruits to be gathered together. Plants were selected for higher yield, dependable flowering, and better performance under local rainfall or temperature. A useful collection of these changes is sometimes called a domestication syndrome. The word syndrome here means a group of traits that often appear together during domestication. Larger fruit, stronger attachment, and reduced natural dispersal make the crop easier for people to manage. They can also make it more dependent on human care. A heavy bell pepper is poorly suited to being swallowed by a bird. A fruit that refuses to drop may leave its seeds trapped on the plant unless someone harvests it. Cultivated peppers therefore gained useful agricultural traits while losing some features that had supported independent life in the wild. Pungency remained only one part of the process. Farmers also selected aroma, wall thickness, fruit position, storage quality, and the time required to ripen. Local foods shaped the plants, while soil and climate limited which plants survived well enough to be selected again. The 5 domesticated species preserve traces of those long regional relationships. Their genomes show inheritance, crossing, and separation across many generations. Living plants can reveal how lineages are related, though they cannot show exactly how a pepper was ground, mixed, or cooked thousands of years ago. Those details survive in smaller traces, including starch grains pressed into the surfaces of ancient tools. Soft fruit rarely survives long enough to become an obvious archaeological object. A chili left in warm soil breaks down quickly. Its skin collapses, the flesh disappears, and most of its color is lost. Stone tools, bones, and hard seeds can remain for thousands of years, while the softer parts of a pepper usually vanish. That makes early chili use difficult to trace through whole fruits alone. Some evidence survives at a scale too small to see without a microscope. Plants store energy in the form of starch. Inside their tissues, that starch is packed into tiny granules, with shapes influenced by the species that produce them. A chili starch grain can carry a particular outline, surface pattern, and central feature. These details allow archaeologists to compare ancient samples with modern reference collections made from known plants. The process begins with an old tool or vessel. Researchers may rinse or gently scrape a small area of its surface, then examine the collected material under magnification. Microscopic grains trapped in cracks can remain long after the meal itself has disappeared. Identification depends on several clues working together. The shape must match known chili starch. The archaeological layer must be securely dated. The tool's location and the other plant remains found nearby must also make sense. A faded red mark by itself would provide very little certainty. The strongest evidence comes from repeated patterns found in clear contexts. Chili starch grains have been reported from 7 archaeological sites spread across a broad part of the Americas. The sites range from the Bahamas and parts of Central America to southern Peru. Some of the evidence dates to roughly 6,000 years before the present. That places chili use thousands of years before European ships began carrying pepper seeds across the Atlantic. At several sites, chili residues appeared beside traces of maize and other cultivated plants. This suggests that peppers were already part of settled food systems, rather than being occasional wild berries gathered without preparation. The residues can show contact with a grinding surface or cooking vessel, though they cannot reveal a complete recipe. A person may have crushed fresh fruit, ground dried peppers, or mixed them with maize. Heat and moisture could have changed the starch before it became lodged in a tool. Archaeologists study those changes carefully, because cooked starch often swells, cracks, or loses part of its original shape. This can help distinguish raw plant material from food that was processed. The tools themselves also provide clues. A grinding stone with worn, polished areas shows repeated use. A vessel with starch trapped along its inner wall may once have held a mixture thick enough to leave residue behind. When chili and maize appear together, the evidence points toward meals in which cultivated crops were combined. The exact flavor, texture, and level of pungency remain unknown. The physical traces still place peppers directly within the work of cooking. These findings also show that chili history belongs first to Indigenous American communities. Farmers selected plants over generations. Cooks learned how different fruits behaved when dried or crushed. Seeds moved between valleys, coasts, islands, and highlands through exchange networks that existed long before European arrival. The plants spread within the Americas already involved trade, migration, and local adaptation. A pepper grown in one region could be carried as dried fruit or seed to another, where farmers selected it again under different rainfall and soil. Microscopic evidence gives that long history a material form. A starch grain may be smaller than the width of a human hair, yet it can remain pressed into stone for thousands of years. Under a microscope, it becomes proof that a particular plant touched a particular object in a particular place. The fruit itself has disappeared, while part of its structure remains. By the time written accounts appeared, chili peppers had already been cultivated, prepared, and exchanged across large parts of the Americas for many generations. Their later global journey began from that deep base of knowledge. A few dry seeds could fit easily inside a pouch or ship's container, and once they entered ocean trade routes, the plant moved into nearly every warm farming region on Earth. When European ships reached the Americas near the end of the 15th century, they were already searching for a different kind of pepper. Black pepper came from the vine Piper nigrum, a tropical plant native to South Asia. Its dried berries had been traded across long distances for centuries and were valuable enough to shape routes between Europe and Asia. The American chili belonged to the genus Capsicum and shared no close botanical relationship with that vine. The familiar name survived because both plants produced a sharp, forceful sensation in food. This was a connection based on taste and trade rather than plant ancestry. Chili seeds began reaching the Iberian Peninsula during the late 15th and early 16th centuries. They were small, dry, and easy to carry. A pouch could hold enough seed to begin a large planting, while a living plant required far more space and care. Once planted in Spain or Portugal, peppers could produce another generation within a single growing season. That gave them an advantage over spices that had to be harvested far away and shipped as finished goods. Portuguese trade routes carried chilies along the coasts of Africa and into the Indian Ocean. Ships linked Brazil with ports in West and East Africa, then continued toward India and Southeast Asia. From those regions, peppers moved again through local markets, farming communities, and older trade systems that already connected Arabia, South Asia, and China. Spanish networks carried plants through the Caribbean and Europe, while later voyages moved them across the Pacific through the Philippines. The spread followed many overlapping paths. A pepper introduced at one port could travel inland by caravan, move along a river, or pass from one village to another through seed exchange. Merchants carried dried fruit. Farmers saved seeds from the plants that performed well. Migrants and displaced communities brought familiar ingredients into new settlements, adding further roots to the plant's movement. Within a few centuries, chilies were being cultivated across much of tropical and subtropical Africa, Asia, and southern Europe. Their rapid success depended partly on climate. Many warm regions already used farming systems suited to tomatoes, eggplants, and other members of the nightshade family. Chilies could grow in garden plots, household courtyards, or larger fields. Some varieties also performed well as annual crops in cooler regions, completing their life cycle during the warm months before frost returned. The crop offered a local source of pungency. Farmers no longer needed to depend entirely on imported black pepper to add a sharp sensation to food. A handful of chili seeds could produce many fruits, and each fruit contained more seed for the following year. This made pungency reproducible within the community. The plant also responded quickly to selection. A farmer in a humid coastal region might save seed from plants that resisted rot. Someone in a cooler upland area could favor early ripening fruit. Cooks influenced those choices by preferring certain colors, levels of heat, or wall thickness. Modern genetic studies have traced parts of this history through more than 10,000 pepper samples preserved in gene banks. A gene bank is a collection that stores seeds or living plant material, so varieties can be studied and protected. DNA patterns within those collections show several major expansions, rather than one simple line moving outward from the Americas. Some groups reflect Portuguese connections among Brazil, Africa, India, and China. Others preserve signs of movement through the Mediterranean, Eastern Europe, and overland Asian trade. Repeated crossing and local selection added new layers after each arrival. East Asia became a major center of pepper diversity. South Asian farmers developed many forms suited to drying, pickling, and fresh use. African regions selected peppers for local climates and food traditions. Eastern European growers produced varieties adapted to shorter seasons and cooler nights. These secondary centers now contain forms that developed far from the plant's original American range, even though their deeper ancestry still leads back across the Atlantic. A pepper could therefore become deeply traditional within a region in only a few hundred years. Its seeds entered local soil, and each harvest changed the population slightly. Drying altered storage. Smoke added new aromas. Fermentation changed acidity and texture. Regional cooking then guided which plants were saved and planted again. The ocean voyage moved the crop into new continents, while the work done after arrival created the peppers people now recognize as their own. Once peppers entered local kitchens, preservation often determined which varieties became useful. Fresh fruit contains a large amount of water and can soften within days after harvest. Thin-walled peppers dry more readily because moisture has a shorter distance to travel from the inner tissue to the surrounding air. Sun-drying, warm ovens, and later mechanical dryers allowed harvest to remain usable long after the growing season ended. Removing water also reduced weight, making dried peppers easier to store and transport. The chemistry became more concentrated at the same time. A dried pepper contains less water per gram, so its pigments, sugars, acids, and capsaicinoids occupy a larger share of the remaining material. Grinding then spreads those compounds through a powder that can be measured by the spoonful. Paprika developed from this combination of drying, grinding, and plant selection. Farmers in parts of Central and Eastern Europe favored peppers that produce strong color and dependable yields. Some varieties remain sweet, while others retain mild or pronounced pungency. Their value depended heavily on the red pigment released into soups, sausages, and stews. That color appears during ripening. Green peppers contain abundant chlorophyll, the pigment plants use to capture light. As many red cultivars mature, chlorophyll declines and a different group of pigments accumulates. These compounds are called carotenoids. Capsaicin and capsaicin provide much of the deep red color associated with ripe peppers and paprika powder. Ripening changes more than appearance. A green jalapeno and a red fruit, left on the same plant, come from the same cultivar, yet their chemistry differs. The ripe fruit usually contains more sugar and a changed mixture of aroma compounds. Its chlorophyll has faded while red pigments have increased. Capsaicin may be present in both stages, though the surrounding flavor is different. Capsaicin itself contributes very little aroma. It creates pungency by activating sensory nerves. The fruity, grassy, floral, or earthy character of a pepper comes from other molecules that travel into the air and reach smell receptors. This separation allows two peppers with similar heat to taste strikingly different. Processing widens those differences. Smoking exposes fruit to volatile compounds released as wood heats and burns. Volatile means able to enter the air easily. Some of these molecules settle on the pepper's skin and move into the tissue, adding aromas associated with toasted wood and smoke. The dried, smoked jalapeno known as chipotle shows how one harvested fruit can acquire a distinct identity through processing. Oil changes the experience in another way. Capsaicin and many aroma compounds mix more readily with fat than with water. Heating dried chili in oil draws these substances outward and distributes them through the liquid. Chinese chili oils use that property to carry pungency and aroma across noodles, vegetables, and dumplings. The oil becomes part of the flavor system rather than a simple container. Fermentation relies on living microbes. In a salted pepper mixture, selected bacteria consume sugars and produce acids. The rising acidity slows many organisms that cause spoilage. Microbial activity also creates new aroma compounds and changes the texture of the fruit. Fermented sauces and pastes therefore taste different from fresh peppers, even when they began with the same harvest. Regional traditions developed through practical choices like these. Korean gochugaru depends on peppers suited to drying and flaking. Piri-piri sauces use small pungent fruits that perform well in parts of Africa. Indian cuisines adopted many dried forms that could be ground, fried in oil, or added whole during cooking. Southeast Asian fresh pastes often use peppers whose aroma remains vivid when crushed with moist ingredients. Each use placed pressure on the crop itself. Cooks preferred fruits that dried evenly, released strong color, or fit easily into a mortar. Farmers saved seed from plants matching those needs. Peppers intended for stuffing grew larger and developed thicker walls. Pickling favored firm fruits that held their shape in acid and brine. Fresh sauces rewarded bright aroma and high juice content. Over generations, repeated seed choice changed the next crop. Processing transformed the harvested fruit, while selection transformed the plants that would produce future harvests. Together, those forces turned a recently introduced species into an ingredient closely fitted to local weather, farming methods, and food. The same process eventually altered nearly every visible feature of the pepper, including whether the fruit burned at all. Place a wild chili berry beside a modern bell pepper, and the effect of human selection becomes immediately visible. The wild fruit may weigh less than a gram. A large bell pepper can weigh more than 200 grams and contain a thick wall of crisp tissue surrounding a broad hollow center. Both can belong to the same species, Capsicum annuum. Jalapenos, poblanos, and many cayenne peppers also belong to that species. Even though their fruits differ greatly in size, shape, color, and pungency. Those differences accumulated because people repeatedly saved seed from plants with useful inherited traits. A larger fruit provided more food from each harvest. Thick walls made peppers crisp enough for fresh eating and sturdy enough to hold fillings during cooking. Broad internal space became valuable in varieties selected for stuffing. Thin walls served a different purpose by allowing moisture to escape quickly during drying. Harvesting also changed the way ripe fruit remained on the plant. Many wild chilies detach easily when a bird pulls at them, or when the fruit reaches full maturity. Domesticated peppers often stay attached more firmly. That stronger connection gives farmers time to collect ripe fruit before it falls onto damp soil, where insects or microbes may reach it. People also selected plants that ripened within a more predictable period, making larger harvests possible at once. Color could be chosen at 2 stages. An immature pepper might remain pale green, deepen toward purple, or begin nearly white. Later ripening could produce red, orange, yellow, or brown fruit, depending on the pigments allowed to accumulate. Farmers preserve colors suited to local markets and food preparation, gradually increasing the frequency of the genes behind them. Pungency could change just as dramatically. Sweet peppers usually carry inherited changes that interrupt the capsaicinoid pathway. Some affect genes required to build capsaicin in the placental tissue. When an essential step no longer functions, the fruit can retain its familiar hollow structure and broad walls, while producing little or no chemical heat. The bell pepper therefore remains recognizably a chili fruit, even though its TRPV1 activating compounds are largely absent. Other farmers moved selection in the opposite direction. Seeds came from plants whose fruits produced reliable pungency, alongside useful flavor or strong yields. Some varieties were kept thin-walled because they dried evenly. Others were selected for fruity aromas, compact shapes, or survival under cool nights and short growing seasons. Heat remained one trait within a larger agricultural package. The material available to breeders came from ordinary biological processes. Mutation introduced new changes in DNA. Recombination shuffled inherited versions of genes when pollen and egg cells formed. Crossing plants brought useful traits together in their offspring. Seed saving then increased the proportion of plants carrying the desired combination. Artificial selection describes this repeated human choice. Its raw material still comes from inheritance and variation already present within plant populations. Traditional farming often preserved that variation in land races. A landrace is a locally maintained crop population containing more genetic diversity than a highly uniform commercial hybrid. Plants within one landrace may differ slightly in fruit shape, ripening time, or disease tolerance. That variation can help the crop continue under changing weather, poor soil, or newly arriving pests. It also gives later breeders more traits to work with. Commercial hybrids are often more consistent. Their plants may produce fruit of nearly identical size and mature within a narrow window. That uniformity simplifies packing and sale, though it can leave less variation within the field. Across both systems, the contrast between wild berry and bell pepper reveals the cumulative effect of many small choices. People altered fruit weight by well over 100-fold. They strengthened attachment, changed wall thickness, and redirected pigment development. The same species could become sweet enough for raw slices or pungent enough to dominate an entire dish. As heat became a deliberate breeding target, everyday words lost precision. One person's mild pepper could feel severe to someone else, and fruit from neighboring plants could vary. A sensation shaped by nerves and experience needed a repeatable method of measurement. In 1912, the American pharmacist Wilbur Scoville proposed a way to compare pepper heat more consistently. The practical problem was simple. People could agree that one extract felt hotter than another. Yet personal sensitivity made precise comparison difficult. Scoville called his method an organoleptic test. Organoleptic means evaluated through the senses, such as taste, smell, or touch. Pepper extract was mixed into sweetened water and given to a tasting panel. The liquid was diluted again and again until the panel could no longer detect a clear burning sensation. A pepper requiring greater dilution received a larger rating. If an extract had to be weakened far more before the burn disappeared, it contained more pungent material. The resulting values became known as Scoville heat units. This method gave farmers, manufacturers, and researchers a shared language for comparing peppers. It still depended heavily on human bodies. One taster might detect capsaicin at a concentration another person barely noticed. Repeated samples could leave the mouth temporarily desensitized, making later extracts seem weaker. Fatigue also mattered, because tasting strong solutions over and over changed attention and comfort. The sweetened water created a standardized setting, though it could not remove every difference among people. Modern laboratories usually measure pungency with an instrument rather than relying on repeated tasting. The most common method is high-performance liquid chromatography, often shortened to HPLC. Chromatography is a way of separating the chemicals in a mixture. A prepared pepper sample is dissolved and pushed through a narrow column packed with special material. The compounds travel through that column at different speeds because each interacts differently with the material inside. Capsaicin may leave the column at one time, while dihydrocapsaicin arrives at the detector slightly later. The detector records a signal as each compound appears. The size of that signal allows the laboratory to calculate how much of each capsaicinoid was present in the sample. Those measured concentrations can then be converted into approximate Scoville heat units. The conversion keeps the familiar scale while replacing the tasting threshold with chemical data. A bell pepper usually falls near zero because it produces almost no capsaicinoids. Jalapenos commonly reach into the thousands. Habaneros can rise into the hundreds of thousands. The strongest cultivated peppers extend into the millions. Those numbers describe chemical concentration. They do not measure aroma, sweetness, texture, or overall flavor quality. They also cannot predict the exact pain every person will report. Individual sensitivity still shapes the experience once the pepper is eaten. Sampling creates another important source of variation. A measurement from one fruit may differ from the average of 20 fruits harvested from the same field. Maturity matters because capsaicinoid production changes as the fruit develops. Fresh weight testing includes the water inside the pepper. Dry weight testing removes that water, making the remaining compounds appear more concentrated per gram. The placental tissue may also produce a much higher reading than a sample taken mostly from the outer wall. Laboratories must therefore record which tissue was tested, how many fruits were combined, and how the material was prepared. Different extraction methods or conversion formulas can shift the final result as well. A published number becomes most useful when the sampling method is clear. The scale gives a repeatable estimate of pungency, rather than a permanent label attached to every fruit of a variety. Weather, plant genetics, and ripeness can move individual samples above or below the familiar range. Even with those limits, instrumental testing changed pepper breeding. Researchers could compare offspring without asking a tasting panel to endure every sample. Growers could identify plants carrying unusually high concentrations and use them in later crosses. Once pungency became a laboratory value, heat could be selected with the same deliberate attention given to yield or fruit size. Breeders soon began treating the upper end of the scale as a record waiting to be surpassed. Breeders now had a numerical target they could carry from one generation to the next. The process begins by choosing parent plants with useful inherited traits. Pollen from one flower is transferred to the stigma of another, allowing genes from both parents to enter the developing seed. The resulting plants may combine strong pungency with a desired aroma, unusual shape, reliable yield, or tolerance for local weather. Their first offspring are only a beginning. Genes are reshuffled again when those plants produce seed, so the following generation may contain fruits with widely different qualities. A breeder may raise hundreds of seedlings, test the peppers, and keep seed from only a small fraction of the population. That cycle continues across several generations. As the chosen traits become more predictable, the line is described as stable. A stable cultivar produces offspring that resemble the parent closely enough for growers to know what kind of fruit the seed is likely to yield. One unusually hot fruit from an early cross cannot establish that consistency by itself. The trait must return through repeated planting over many seasons. During the early 21st century, this work pushed measured pungency steadily upward. Bhojolokia, associated with northeastern India, became widely known after testing placed it above 1 million Scoville heat units. Trinidad scorpion types later carried the upper range higher. These peppers helped establish a breeding movement in which capsaicinoid concentration became a carefully measured target. The Carolina Reaper followed. developed by American breeder Ed Curry. It combined extreme pungency with a deeply wrinkled surface and a pointed tail. Its reported average reached about 1,640,000 Scoville heat units. Individual samples could vary around that figure because the average came from tested peppers across a population. Curry's breeding program eventually produced Pepper X. He spent more than a decade crossing selected plants and growing their descendants through repeated cycles. Only lines that continued producing unusually high capsaicinoid levels were carried forward. Winthrop University in South Carolina tested specimens collected across 4 years. The reported average was 2,693,000 Scoville heat units. As of June 2026, Guinness World Records identifies Pepper X as the official hottest chili pepper. That figure describes the tested average. A fruit harvested from one branch may fall above or below it. The record comes from controlled sampling across multiple harvests and laboratory measurement of capsaicinoid concentration. Superhot cultivars often have unusual internal structures. Their walls may fold inward and outward, producing the heavily wrinkled surface seen on many record-level peppers. Placental tissue can spread along those folds, increasing the internal area containing cells able to produce capsaicinoids. Wrinkling alone cannot prove that a pepper is extremely pungent. Mild fruits can also develop uneven surfaces. The amount and activity of capsaicinoid-producing tissue provide the more important connection. Human selection expanded this natural defense through ordinary inheritance. Crosses reshuffled gene versions already present in the parent plants, while laboratory measurements helped breeders identify promising offspring. Saving seed from those plants over many generations made the desired combination increasingly dependable. The final fruit may look unlike a small wild chili, though its extreme chemistry arose through the same biological processes that once enlarged a berry into a bell pepper. The original warning molecule had become a designed agricultural trait, measured in millions. Even a carefully stabilized cultivar cannot produce an identical reading in every fruit. Its genes establish a range of possibilities. Sunlight and water supply influence development, while temperature, maturity, plant health, and fruit position help determine how much capsaicin finally accumulates. Two peppers hanging from the same branch can carry noticeably different levels of heat. They share the same parent plant and nearly the same genetic instructions. One fruit may sit closer to the stem, partly shaded by leaves. The other may receive stronger sunlight, develop more quickly, or remain on the plant several days longer. Those small differences can change the chemistry forming inside each fruit. Genes establish the broad capacity for capsaicinoid production. They determine whether the required enzymes are present and how the placental tissue develops. The final concentration still depends on how strongly those inherited pathways operate during fruit growth. This relationship is called genotype by environment interaction. Genotype means the inherited genetic makeup of the plant. The environment includes surrounding conditions such as temperature, water supply, and soil nutrition. The two influence each other, so the same growing condition can produce different results in separate cultivars. Capsaicinoid concentration also changes with maturity. A newly formed pepper contains very little pungent material. Production rises as the fruit expands and its internal tissues develop. The highest concentration may occur before or near full ripeness, depending on the cultivar. Later changes can include continued production, leveling, or partial breakdown. Harvesting two fruits at different stages can therefore produce different laboratory readings, even when both appear ready to eat. Temperature affects the enzymes building capsaicin. Warm conditions may support strong production within a suitable range. Excessive heat can interfere with flowering, pollen function, and fruit development. Cool nights may slow growth or delay ripening. Each variety responds according to its own inherited tolerances, which is why one temperature rule cannot describe every pepper. Water limitation is equally complicated. Some experiments have found increased capsaicinoid concentration under moderate water stress. Other cultivars show little change or a decline. Timing matters because restricted water during flowering may reduce the number of fruits that form, while stress later in development affects fruit size and chemistry differently. Severe drought can leave the plant with fewer leaves, smaller peppers, and a reduced total harvest. A surviving fruit might contain more capsaicin per gram, while the field produces less usable pungent material overall. Nutrition shapes the resources available for growth. Nitrogen supports leaves and proteins, though excessive nitrogen can encourage leafy growth at the expense of fruiting. Potassium contributes to water balance and fruit development. The effect of any fertilizer depends on the soil, the cultivar, and the amount already available to the roots. Fruit load creates another division of resources. A plant carrying many developing peppers must distribute sugars and minerals across the entire crop. A plant with fewer fruits may direct more material into each one. Position matters as well because leaves near a fruit can supply much of the sugar used by that developing tissue. Disease can interrupt the process at almost any stage. Anthracnose fungi produce sunken lesions on ripening fruit. Viruses may distort leaves and reduce photosynthesis. Root pathogens interfere with water and nutrient uptake, while fruit rots can destroy peppers before harvest. A variety bred only for extreme heat would have little agricultural value if it produced poorly or lost most of its fruit to disease. Farmers therefore balance pungency with yield, shelf life, flavor, and climate resilience. Drying adds a final source of confusion. A fresh pepper contains substantial water. After drying, the same capsaicinoids remain in a much lighter piece of material. The concentration per gram rises mainly because water has been removed. That does not mean the drying process created an equal amount of new capsaicin. A record value remains useful when the sample and testing conditions are known. The number describes a measured fruit or group of fruits, rather than every pepper the cultivar will ever produce. Living plants respond continuously to development and surroundings, placing each fruit somewhere within an inherited range. Researchers gain greater control by removing capsaicin from that variable fruit and preparing known concentrations, turning the pepper's chemical warning into a precise tool for studying pain. With purified capsaicin, researchers could choose the concentration, control the area of exposure, and watch a specific group of sensory nerves respond. That precision helped identify the receptor now called TRPV1. In 1997, scientists used capsaicin while isolating the gene that produces this heat-sensitive channel. The work connected the familiar burn of a pepper to a physical protein embedded in sensory nerve cells. TRPV1 occurs especially often in nociceptors. These neurons detect conditions that may damage tissue, including intense heat and irritating chemicals. When capsaicin reaches the receptor, the channel opens and allows charged particles to enter the nerve ending. The cell becomes strongly excited and begins sending pain signals. A concentrated medical exposure, therefore, produces an initial period of pronounced burning. The response may also involve redness as nearby blood vessels widen. Continued activation changes the nerve ending itself. Large amounts of calcium enter through open channels, placing the cell under temporary chemical stress. Signaling molecules are depleted, and parts of the nerve ending become less able to transmit repeated warnings. Doctors call this defunctionalization. The nerve remains present, though its exposed ending functions less effectively for a period of time. This effect differs from permanently destroying every nerve in the treated skin. Recovery can occur gradually as the sensory ending restores its normal structures and signaling ability. Medicine uses that temporary reduction to treat certain forms of neuropathic pain. Neuropathic pain comes from damaged or abnormally active nerves. In such conditions, sensory fibers may continue sending warnings after the original injury has healed, or when no new tissue damage is occurring. Carefully controlled capsaicin exposure can quiet some of those overactive fibers after the intense opening phase has passed. One regulated example in the United States is Catenza, an 8% capsaicin topical system. 8% is far more concentrated than ordinary over-the-counter capsaicin creams. Its approved adult uses include neuropathic pain associated with postherpetic neuralgia, which is lingering nerve pain after shingles. The other approved use is neuropathic pain associated with diabetic peripheral neuropathy of the feet, where diabetes has damaged sensory nerves. Healthcare professionals apply and remove the treatment. Protective gloves are used because accidental transfer can strongly irritate skin or eyes. The treatment area is carefully selected, and ventilation helps prevent airborne capsaicin from bothering the nose or airways. Placement is restricted to appropriate intact skin, rather than sensitive surfaces. Clinical control also allows staff to manage the temporary rise in discomfort and monitor the patient during treatment. Eating chili peppers does not reproduce this therapy. A meal delivers an uncertain dose across the mouth and digestive tract, while the medical system places a measured concentration on a defined area of skin. Observational studies of chili consumption sometimes explore broader links with health, though those questions remain separate from the established use of regulated topical capsaicin for specific nerve pain conditions. The molecule has also remained valuable in laboratories. Researchers can activate TRPV1 to identify pain-sensing fibers, examine inflammation, and study how temperature information enters the nervous system. A chemical once shaped by interactions among peppers, fungi, and animals became a precise experimental tool because its target receptor could be controlled and observed. The plant still has more to offer beyond capsaicin. Gene banks preserve seeds from cultivated varieties, wild relatives, and locally maintained land races. Those collections contain inherited differences affecting resistance to fruit rots, viruses, and root diseases. Other plants carry traits connected to drought tolerance, cool weather growth, aroma, wall thickness, or ripening time. A landrace that looks uneven beside a commercial hybrid may hold a useful gene that has survived generations of farming under difficult local conditions. Plant breeders can cross that material into future crops, then measure the offspring and retain useful combinations. This work may help peppers remain productive as growing regions face changing rainfall, new pathogens, and warmer seasons. The same long history that intensified the burn can therefore support disease resistance and climate adaptation. From a purified medical dose back to a seed stored in a gene bank, capsaicin remains connected to the living fruit that first assembled it. The ordinary pepper can now bring the full story together: receptor, animal, fungus, farmer, laboratory, and medicine, all linked by one inherited chemical pathway. Return to the pepper on the cutting board. Its outer wall is smooth and cool. Inside, pale placental tissue holds the cells that built its capsaicinoids, while flat seeds rest nearby in the hollow chamber. The surface does not glow. Its temperature matches the room around it. The power begins when the molecule reaches a living receptor prepared to read it. That meeting happens quickly. Within milliseconds, capsaicin binds to TRPV1 and opens a channel through the nerve membrane. Sodium and calcium ions move inward. The nerve produces an electrical signal that travels toward the spinal cord and brain. A plant chemical has become nerve activity. The body answers with actions shaped for protection. Saliva increases. Tears may form, while sweat can gather across the face as surface blood vessels widen. These responses belong to systems that wash exposed tissue and release heat. Their activity is genuine, even though the pepper has not raised the body to a dangerous temperature. The fruit required far longer to prepare that signal. Over several weeks, its tissues expanded from a fertilized flower. Inherited enzymes assembled the capsaicinoids in specialized placental cells. Water supply, temperature, maturity, and the plant's health influenced how much accumulated before harvest. Another fruit on the same branch could finish with a different concentration. Across generations, those differences became material for selection. Wild peppers that discouraged seed-crushing mammals could leave more intact offspring. Birds experienced capsaicin through a differently structured TRPV1 channel and often swallowed the small red fruits whole. Their movement carried seeds away from the parent plant. Piercing insects created openings for fusarium and other fungi, adding pressure from a much smaller enemy. Capsaicinoids slowed some fungal growth, while the cost of producing pungent fruit kept milder plants useful in certain dry environments. Human selection entered this system thousands of years ago. Indigenous communities across the Americas gathered peppers, moved their seeds, and cultivated plants with useful fruit. Repeated choices enlarged some fruits and strengthened their attachment to the stem. Further selection changed wall thickness, color, aroma, and ripening patterns. Other choices preserved pungency or removed most of it. A small wild berry gradually gave rise to forms ranging from sweet bell peppers to thin-walled drying chilies. After the late 15th century, portable seeds moved through maritime and overland trade. Peppers reached Europe, Africa, Asia, and new islands within a few centuries. Farmers in each region selected plants for local weather and familiar methods of cooking. Drying concentrated the remaining material. Smoking deposited volatile compounds. Oil redistributed fat-soluble molecules. And fermentation allowed microbes to reshape sugars into acids and new aromas. The same genus became part of paprika, chili oils, fresh paste, and many other regional foods. Measurement extended the process. Wilbur Scoville's tasting method turned perceived pungency into a dilution scale in 1912. Modern laboratories separated capsaicinoids with HPLC and converted chemical concentration into Scoville heat units. Artificial selection then stretched the trait from values near zero to tested averages above 2 million. The warning molecule had become a breeding target with a number attached to it. Medicine completed another part of the conversion. Purified capsaicin helped researchers identify and study TRPV1. In controlled high doses, intense receptor activation can temporarily reduce the ability of some pain-sensing endings to transmit abnormal signals. The pathway responsible for the initial burn could then serve carefully defined medical purposes under professional care. All of these consequences remain connected to the fruit lying quietly on the board. Plant metabolism assembled the molecule, while animal receptors determined how it was experienced. Ecological interactions influenced which peppers reproduced successfully. Human learning turned a temporary alarm into a valued sensation. Agriculture preserved that preference in seeds, and trade carried it across continents. The chili contains information assembled by a plant and interpreted by a nerve. Evolution filtered its effects through animals, insects, and fungi. Human history transformed the same information into flavor, measurement, agriculture, and medicine. The cut pepper remains cool and still. Its invisible molecules wait quietly for a living system capable of reading their message.